Welcome. Thanks for coming out. Thanks to all you folks who came out in person, to you Zoom folks online, and maybe you folks that are watching at a later date. Um, so my name's Dustin Rubenstein. I run the Advanced Genome Editing Lab. I also manage the Bio Garage. And I'm here to talk to you today because we kind of have a theme going for this session. And that theme is how can we help you researchers enable your work um, by providing sort of end-to-end workflows, right, from beginning to end. That's kind of the theme of what we're talking about today. So I'm going to tell you a little bit about the Bio Garage here at the Biotech Center. That will help you in that respect. And then we're lucky to have a couple of experts from Thermo Fisher who are instrument experts who can really help you. We can really drill in details in a couple cool instruments, and we'll get to that in a second. Um, so, um, Starting with the BioGarage. What is a BioGarage? The idea for the BioGarage was always to help close that last gap of maybe an experimental need you have, but you just don't have the resources. That resource might be access to instrumentation, it might be some sort of workspace, it might be the expertise to run it. So there's kind of three things that the BioGarage offers. Number one is direct access, walk-up access to instrumentation. Really great modern instrumentation for you. Another is workspaces that allow you to get the things done that you need, including some specialized workspaces like BSL2 spaces, and we'll talk about that quickly. And also we can just handle, the BioGarage can just simply handle the workload for you and just do some of these experiments to free you up to do the other more specialized things you need to do. All right, so first off, how do we get to the BioGarage? So BioGarage gets by in a system of memberships. So there are both single memberships and full lab memberships. And I encourage you to check the website. You'll see that the membership costs are very, very reasonable, and it really only takes at this time two individual memberships to get you access the same for the price of two individual memberships, your whole lab can get a lab membership as well. Um, well, what do you get for that? You have uh, unlimited free access to tons of instruments that are in there, and unlimited access to some paid instruments as well as access to sorry, as well as access to workspaces, specialized workspaces. So let's talk a little bit about the free instrumentation that's available to you. Some staples, things that just every molecular lab needs, PCR blocks. So part of the idea here is that yes, we have access to cutting edge instrumentation. That's cool. We also have access to just the molecular biology tools that are standard in some labs, but other labs may not have. So if you're if you're in a molecular lab that's kind of just starting up, rather than having to reinvent the wheel and buy all these kind of rote things, the idea is that the BioGarage is a workspace for you to get your stuff done. So it starts with essential things like PCR blocks um, uh, and then all kinds of things, sample characterization. So whether it's qubit for concentration, the tape station e-gels. So tape station, for example, 96 wells, essentially gel electrophoresis for 96 wells automated. E-gels are great. They're super cost-effective gels. You just pop your gel in the machine, and you can load up to 96 samples, as well as a nanodrop. Things for sample prep like bead homogenizer. So the point of here is that all of these are, if you're a BioGarage member, you can just come up and use these right now as much as you need. You need to bring your own supplies, but these are available to you. Additionally, there's some fee per use instrumentation. So some of the exciting things here, we've got both digital, so Thermo Fisher's QuantStudio Absolute Q for digital PCR, which is a really exciting instrument. And the Thermo Fisher QuantStudio Pro 7, we actually have a few of those in the lab. Those are also exciting instruments. Allows you to really high throughput, get through tons of samples in a cost-effective manner. But you're going to hear a lot more about that later, so I'm going to hold off on that. Susan Hetzel is going to be talking more about that. And then we got, bioanalyzer. We have NextSeq available for special requests. And then, of course, the Kingfish Apex, which we're lucky that Christian is going to come and talk to us about. But it's, the idea of really high throughput RNA, DNA preps of 96 samples at a time, really empowering your research. So again, the idea of, complete workflows, right? So if you have a BioGarage membership, you need zero capital expenses. You've got everything you need to do to run your samples and collect data. I think, really want to sort of emphasize that. Everything from sample prep to understanding what's in your sample to data acquisition. The BioGarage. So that's kind of the idea of BioGarage instrumentation. Also, you have available specialized workspaces, right? I mean, you may have a lab. In some cases, you don't, but you may be missing certain specialized lab spaces. For example, culture BSL2 enabled space. We have another microbial room that allows risk group 2 microorganisms. So if these are things that are of interest to you, we have these spaces that are already off. Of course, we're going to have to we have to make the proper precautions, and we'll discuss together, what the experiments are and what the limitations are. And but you have these spaces available to you today. They're spaces you need. And then I want to push kind of the third idea here, BioGarage as end-to-end services. That certainly, yes, there's some experiments that could be done in your lab in some cases, but my guess is if you're like, a lot of other grad students or postdocs, you probably have more than like a couple experiments to do, and it'd be nice just to, unload those. The idea here with BioGarage is that it's services like qPCR, digital PCR, those are things that you shouldn't really need to do. So we've set up systems with automation, and we set up really cost-effective methods for you to rather than sort of duplicate these efforts, just sort of hand them off to the BioGarage, and you can run these data, give us samples and hand it back to you. That covers things like qPCR and digital PCR, which you might use for gene expression, CNV genotyping. We also can help with things like plasmid preparations, whether you need, plasmid scale-ups, or, we can get you a glycerol stock for archiving you can toss in your -80. The idea here is that I think there's kind of two ideas here. Number one is that for some labs that have the expertise, they might have the expertise, but just be busy. So they can just kind of hand it to us and you can get your own stuff done. And the other thing is that there may be labs that are, understaffed or just don't really have people who run qPCR, in which case there's no reason this day and age that should hold you back. You have someone like A that can help you with that. Ignore that image for now. So for example, with qPCR services, I duplicated there. These are really reasonable, right? So after a setup cost of 24 samples, each additional sample is only $4. Again, you just hand us a sample. We can help you select your primer probe set, which would be an additional cost, but we can bank that for you and you can always come back to it. And we can run those samples to you and get that data back to you, analyzed and ready. And then I want to finally kind of introduce to you the idea of truly end-to-end services. This is maybe more of a conceptual note than something that is sort of in practice now, but I think there is sort of a modern concept of collaborations on campuses that recognize that that molecular biology, genomics, proteomics, all these biomedical and these research fields are so highly specialized that it's really difficult for one lab to be good at everything. In fact, it's almost more efficient to rely on specialized cores. I think there's a lot of interest sort of at this sort of the bleeding edge of research on campuses, this idea that rather than having individuals in labs actually doing the experiments, they're really more like project managers, right? I mean, you have those in the lab are the content experts. You know what's been done in the past. You know where those critical research gaps are. You know the experiments that needs to be done, the questions that need to be asked. But why go to the next step and actually do the work And you could coordinate it among groups. So the BioGarage, for example, could help facilitate things like, generate full project workflows and help you kind of do that work for you so that you can you can occupy yourself more cerebrally thinking about the bigger picture in those questions and writing papers and analyzing data and understanding what those data mean to your field rather than doing the nitty gritty. Kind of a concept, but it's something that we're starting to hear interest in, where we really just do help shuttle these experiments from beginning to end. Okay, so that's as much as I have about the BioGarage. If there are any questions, I'm happy to answer questions about the BioGarage. Yeah.
Great. I see you have now the bioanalyzer and the tape station in the BioGarage. Will you stock reagents and chips for both of those instrumentation? And if so, which ones?
So as of right now, today, the moment today, bioanalyzer, we do have stocked reagents for those, and that's particularly unique because some of the shelf life of those are fairly short. So rather than buying in bulk, we do have those available. So if you just need one chip, come to us. For the tape station, as of now, as of today, the idea is that it's completely free in walk-up, but you need to bring everything yourself. So just to note, I mean, the most expensive part, of course, there's one reagent course that's the most expensive. There's tapes. There are all kinds of these smaller little ancillary things you need. I think just in the interest of getting our users access, getting you guys access to this instrument, we just want to get it in there and get them access so they can start now. We're happy if it seems like it's really annoying for people to buy these little like ticky-tack little pieces and gizmos. We're certainly considering a model where we just provide those, but there would be some sort of small walk-up fee. And I think it just seems maybe, I'm not sure what's more irritating to our users, small walk-up fees or buying ticky-tack gizmos. So right now we just aired for the fact of just getting it up and running. But yeah, good question.
What's the turnaround time on like the walk-up services?
Yeah, walk-up services. So most everything, so there's some things that are scheduled and some things that aren't. So things that aren't, you could literally just get up out of your chair and just go in there right now and use it. Otherwise, for a lot of the walk-up instruments, you need to schedule it a day in advance. So, if that's your question, or if you're talking about like the runtime for some of these instruments, or yeah, runtime. So, I mean, the QuantStudio, the QuantStudio Pro 7 is what, like a couple hours or hour and a half? Not 40 minutes. Yeah, right. If you run in the fastest mode, it's like 40 minutes. I think digital might be a little longer than that. What's that? Hour and a half. So I mean, so pretty quick. I mean, you get your data really quickly. Yeah, and we send out the native data format as well as a sort of an Excel file that has things a little more meaningfully analyzed, but you get both. Awesome. Good question.
You mentioned that we can submit samples for, say, like automation and whatnot. Is that different from submitting samples like to the, say, like the Gene Expression Center, or is that like something specific, like separate with BioGarage compared to, say, like other services through Biotech?
Yeah, so I guess that's a good point. So I guess what I was referring to is that we have a liquid handler that does great with taking large numbers of samples and converting them into sort of qPCR plates. I think the idea of automation, I can't necessarily, I mean, Sandra, I don't know if you want to speak to that. I know it's something we're always improving.
So where it would differentiate between the BioGarage and the Gene Expression Center, the BioGarage, you can walk up and do your own, let's say, RNA QC. You can use the NanoDrop, you can use the bioanalyzer or the tape station on your own. Versus submitting to the GEC, we perform the QC for you as well as the interpretation. So you have both options available for that. Were you talking in regards to RT-qPCR as well, or just the RNA QC?
I think I can.
I like what you're asking. I mean, if what you're wondering is, can you get access to automation instruments, right? Maybe it's not qPCR, maybe there's a million reasons people use liquid handlers, right? So stay tuned. That's something we're actively working on. I think it's something, if that's something we recognize as a huge need on campus, and we're trying to fight that fight to get access for essentially a liquid handler that would allow you to do whatever it is you need to do. It's not available today, but come talk. If there is a project you want to do today, we'll find a way to make it work. It's not something we have right now on the website as like a click here and buy this, but if there's a project you want to do tomorrow, we'll get it, we'll find a way to get it to work for you. But in the meantime, recognize that as a need. So I appreciate that question. Yeah, I'm still gonna try. Train. Right. Okay. Any other questions? All right. Thanks, everyone. Excellent. Thanks, everyone. Okay, so next up we have Christian Kiss. That's Christian Kiss. He's a field application staff scientist at Thermo Scientific. He brings over 15 years of experience with Thermo Fisher, combining deep technical expertise with a focus on customer success and practical application. In his current role, he serves as senior technical expert and key liaison across customers, sales, R&D, and project management, helping to teach and design and optimize and troubleshoot complex sample preparation workflows and automation solutions for nucleic acid protein purification. That is super helpful. Before joining Thermo Fisher, Christian spent several years at Vanderbilt, conducting research in molecular biology with a focus on cancer signaling. His background includes extensive hands-on experience in qPCR, PCR, cloning, cell culture, protein work. He holds degrees in biology and chemistry from University of Tennessee at Chattanooga, and pursued doctoral studies in molecular biology at Vanderbilt. So with that, forward to hearing what you have to say about the Kingfisher.
Awesome. Thank you. Put that right there. Yeah. Can y'all hear me okay? Perfect. All right. Well, I appreciate that introduction. Yeah, we're going to talk about sample prep and see how it can fit your potential needs in the lab. I guess we already did an introduction, so just my name is Christian Kiss. Nobody ever wants to say my last name, but keep it simple. That's all it is. All right, let's talk about agenda. For today, we're going to break this up into a couple of different sections. First of all, we'll talk about the comparison of purification methods because we're going to focus on the Kingfisher later. But why do we want to do that, and why not go other routes of purification? And I'll walk through those answers. Then I want to focus on what instruments we have as solutions for automation. And then following that, we'll do an Apex walkthrough. Part of that will be running a 5-6 minute demo script so you can see how easy it is to actually get plates onto the instrument and what the instrument is actually doing. So it's a nice visual to get that understanding. Once we finish that, we'll just do some really quick walkthroughs on some nucleic acid options as well as some protein options. This is not meant to be a deep dive today, but just exposure, right? What do we have available that might be of interest to you for doing that purification? And then we always have lots of resources. You can connect back with us at a later time just to get more detailed walkthroughs or any kind of explanation that you might need for specifics. And then hopefully we'll have some time for some Q&A at the end. All right. When we talk about purification, I'm mostly going to focus on nucleic acid purification, just so we understand the basics of that. Most people these days, when they start with purification, they go right to columns. They've been around for a while. They're easy to get started. Unfortunately, they're really not the best way to do purification, and that's simply because of what it actually is. It is simply a plastic tube with a silica matrix in it, right? And when you put sample through, it just flows through, your wash flows through, your elution flows through, but there's no mixing going on, there's no shaking. It's really hard to change what you are actually doing. And depending on your sample, of course, if it's not appropriately homogenized, like tissues and plant material and so forth, well, now you have the potential to start clogging your columns, right? And so there's always issues that can happen. Ultimately, it just means that there's the potential for day-to-day, person-to-person variability with how you are doing that purification. And as scientists, we don't really want that, right? We want consistency and reproducibility in that process. So once you realize columns are not your best option, most people tend to gravitate towards magnetic beads. And it's a little bit different. So instead of this silica tube, you're going to have a silica-coated magnetic bead, right? And so the way it works is you bind your target in solution. Typically, you will have a magnet that will pull those beads down or to the side. And then you just simply use pipetting motion to move those liquids in and out. Now, it's better than columns because you can actually take that tube of material and you can literally shake it for as fast and as long as you want. And logically speaking, it just makes sense. You're enhancing that purification process. You're trying to shake out the impurities. It improves the results that you are going to get. Now, even though it is better than columns, though, there's still one caveat with this, and that is how close do you get that pipette tip to those beads to make sure you're doing a clean liquid transfer? Well, if you're not close enough, you've got a bubble of liquid that's left that stays in there and that continues to carry contaminants forward. On the other hand, if your tip is too close, then, right, you start pulling out some of your beads, and then now you're losing some of your sample. So trying to optimize that can be a challenge for some people. This method can be used on liquid handlers, but it doesn't change the principle of what's going on. It is still using the magnetic beads to bind target. The liquid handler will pull those beads down, and now you're just using a machine to do that pipetting for you. And so trying to optimize that tip can still be somewhat challenging in that process. Well, once you realize you can still go a little bit further, we go to inverse magnetic particle processing, and this is actually what the Kingfisher does. So it still uses magnetic beads, but rather than pulling the beads down, there's actually going to be a magnet on the instrument that is going to be represented in purple color. Typically, we have a tip comb that's represented by blue on the screen here. And those two, when they come together, are going to drop down into the solution to collect all those beads when you're ready to transfer. The magnet is only polarized at the very bottom, so that's the only place you're going to collect beads. They're not scattered all along the side. But once you do collect the beads, they'll be moved over to the next solution. The instrument will remove the magnet from the tip comb. So now, right, no magnetism. Those beads are just going to be left in that solution. And then the instrument will take that empty tip comb and use that as a plunger. So you get this large amount of volume displacement, and that's basically your mixing. It's a lot better than just taking something and shaking it back and forth because that volume plunging is a lot of turnover in that liquid, right? Well, the other thing about the Kingfisher is when you do something like this, it's basically a set of instructions that are going to be executed. And those instructions are going to be the same whether you do it today, tomorrow, next week, or so forth. So as an example, if a particular step in the process is going to be 5 minutes long at a fast speed, well, every time you run it, it will literally always be 5 minutes at that fast speed. And so now, when you look at the other methods, how many times are you pipetting? How long are you shaking? How fast are you shaking? That's all variability that's eliminated by going through an automated process like this, where you are just executing that set of instructions. So now you have that consistency and reproducibility that you need. Well, that's as simple as the instrument gets in that process. You basically let go of your beads, you mix, and then you collect. And as you go through that process, through all the steps, let go, mix, collect, let go, mix, collect, and so forth. And this is what gives you the good results. So when working with beads like this, you have two advantages, typically as good, if not better, purity and yield in the process. And it makes sense because with the purity part, right, we're only moving beads from step to step. So a lot of those liquids are left behind, and that means your contaminants are largely left behind as well. And so the potential to get a better 260-280 ratio, for example, is going to be much better. For the yield part, not only is the mixing and things like that controlled with a set of instructions, but so is the bead collection process. And so there is software that you can go into to optimize that if you need to, just to make sure you're pulling up most, if not all of your beads throughout all those steps. And again, it makes sense if you're not leaving your beads behind, you're not losing any of your sample as well. So this is the best way in terms of methodology to do purification, and this is actually what the Kingfisher Apex does as well. With beads, they offer a few other advantages too. They generally have a pretty large surface area, so you can capture quite a bit of material. To put that into perspective, on the 96 format on the instrument, out of the box quantity of beads and reagents, you can typically collect up to about 20 micrograms worth of DNA, RNA, or total nucleic acid. And that's actually quite a bit of material to do a lot of downstream testing. Now, we do have a variety of magnets just to change those volumes that we are going to be working with, so it's easy to scale up and scale down. And that's the other thing about columns, right? You can't cut a column in half or pour more column in there, so it's very hard to scale. Where with beads, it's a slurry solution. So you just pipette more or less, and you can actually go into the software and simply change the volumes that you are going to be working with in those scripts. It's very easy to do. When we let go of those beads, we slosh them around. It's great for that mixing and for driving out those impurities. Viscosity, you can imagine, is also not really an issue since we're just sloshing the beads around within that liquid. And we do have the software again, so if we need to speed things up or slow things down to enhance that process, that's really easy to do as well. Beads are easy to automate. We put them on the Kingfisher, so that's self-explanatory. And then for low to high throughput applications, looks more at the instrument side of things. Again, let's look at a 96 format magnetic head. That magnetic head is actually a solid piece of neodymium metal, and that is basically a permanent earth magnet, stable for over 100 years. So those pins are all identical. They don't change over the life of the instrument. And that means you can get equivalent results across the plate, right? Well, if you only have 8 samples, you don't have to fill up the entire plate as well. Don't waste your reagents. You can put those in a column, put them in a row, scatter them around the plate. Because those pins are identical and permanently magnetized, well, you don't have to worry about any edge effects, center effects, or just kind of variability in that processing, right? And anything that's empty, like processing air, so it doesn't affect any adjacent samples. The other part, as I already mentioned, with the different magnetic heads, there are different volume ranges for each of those, so it's possible to scale up and down simply through the change of accessories that go with the instrument. You also have to keep in mind bead quality too, though, in conjunction with the instrument. That definitely matters. I know there are a lot of different types of kits and beads out there. Our kits at Thermo Fisher, whether they're nucleic acid or protein beads, they are very high quality beads. They tend to be nice and round, very low background, so very little non-specific binding overall, and that's what we want from the process. But very reproducible, easy to collect, easy to use overall. Other vendor beads might be more popcorn shaped or cauliflower shaped, and you can imagine all those nooks and crannies are actually just going to absorb more material that you don't want. Sometimes beads are really not even beads, they're just flakes, right? And so they can be a little more challenging to work with. So keep that bead quality in mind because overall it will make a difference too. Now, if you're looking to get an instrument, just for sake of knowledge, this is what we have available. We've actually been around since 1999, so Kingfishers are what I call a proven technology, being around for about 27 years at this point. However, we only sell and support the red box items. So we've got the Kingfisher Apex for you. Available in BioGarage, I think you said it was, right? But if you want to get your own instrument, we have other different models too. We'll start with the Kingfisher Flex that came out in 2008. That is an 8-spot turntable. That basically means each spot represents a step in the process. So one plate will be sample, one plate will be a wash, one plate will be a dilution, and so forth. And then there's a magnetic head assembly in the back, and the turntable simply swaps plates out as you are doing your processing. So it's a very nice way to do that high throughput. Now, keep in mind, though, that instrument came out in 2008. In terms of scientific equipment, that thing is ancient, right? About 18 years old. We still sell it and support it. But I would encourage people to look more at the Apex, which came out in 2021. It does everything the Flex does. It's basically the next generation model of that turntable style unit. It does have a lot of extra features, and we'll walk through that in just a sec. We also have an Apex DX. Not sure if this will be relevant here, but it's more of a clinical model. It is our IVD instrument. So if you do qPCR and you have a QuantStudio instrument that is compatible with our Diomni software for the analysis of that, well, our Kingfisher Apex DX can tap into that and keep track of the entire workflow from extraction all the way through purification. If you're not doing that, you don't necessarily need the Apex DX because the regular Apex also has control access functions to lock it down if you need to. A Presto is also high throughput, up to 96 samples at a time, but it is more of an integration model, so it sits inside of a liquid handler. Now you marry the best of both worlds, where you have the liquid handler prepping your plates, and it just feeds things to the Presto to do magnetic bead movement, which we now know is a better way of doing purification. And then if you have low throughput needs, the Kingfisher Duo Prime is exactly that. It's more of an entry-level unit. Instead of doing up to 96 samples at a time, it only does up to 12 samples at a time. All of these work the same way mechanically, so they're very interchangeable in terms of the protocols that they can do. And they also have different volume formats. So the high throughput instruments that go up to 96 samples at a time, there's a higher volume head that's only 24 samples at a time, but it's about 5 times the amount of processing volume that you can have with that. So just for awareness, again, these are the instruments that we have. Anything that is not boxed in red is discontinued or obsolete, which means you'll never get service on it. So don't go looking for that eBay bargain because you might not get it serviced. All right, and lastly, just to close this part out, the instruments also provide very low risk because of the no liquid transfer. That's the main benefit, but also because of the way that it functions. There are going to be certain plastics associated with the instrument. Those are Thermo Fisher Kingfisher branded plastics. We have some examples here that I'll use in a minute. But those are going to have maximum volumes. And if you think about it, when that tip comb plunges all the way down, the liquid level rises. The maximum volume is something we set so that when that tip comb is all the way at the bottom, the liquid still does not reach the top of the plate. So you don't get this spillover and things like that. And that's how we avoid that form of contamination. The other thing the instrument does as well is it looks at the script. It'll look at the volumes for your reagents that are coded, and it takes that information to keep all of that mixing inside that column of liquid. So whether you have 1,000 microliters in a plate or only 200 microliters, the tip comb never leaves that liquid volume. And so this way it doesn't come out, doesn't go back, it doesn't splash, cause aerosolization and foaming. And those are the things that actually lead to cross-contamination as well. So overall, it's a lot of different things that come together to give you a nice process. We already talked about the viscosity and scaling up and down. Automation, you can integrate this with robotics. And then on the low end for concentration, we can actually get to some pretty low volumes for DNA, RNA, protein, whatever it is you're working with. And that's all done through the scripting. All right, we're going to change gears just a little bit and do a quick walkthrough of the Kingfisher. Just one slide to introduce the features, and then what I'm going to do is just start a quick 5-minute protocol and walk you through all the actions. So I mentioned this does everything the Flex does. It's just a more modern instrument. So the extra features are that it can hold multiple heads, multiple heat blocks. This way you have different volume formats and you don't have to manually change those parts. We've added UV decontamination to this. So if you are working with some pathogens or other types of hazardous material, you can decontaminate the unit. If you've got a door, you can run it open and closed. It has to be closed for UV. There's a barcode scanner. That's to scan your plastics and also the parts as you assemble the unit. So now the instrument knows what's on there and what location it is. We can elute into storage tubes as an additional feature on this one. And then we have a really nice touchscreen, large visual format. It's super easy to navigate, literally like a 5-10 minute training on that. You can't get lost, so it's just easy to use overall. We do have reports generating on this automatically. The touchscreen also can give you that controlled access if you just want to lock down the unit and have people log in. And then the touchscreen also has all the full functionality of the unit, so you don't have to have a separate computer. You can do the instrument operation, you can do script writing, you can do part installation and deinstallation and so forth. It's all contained in that user interface over there. All right, well, let's walk through the instrument a little bit. I'm going to change over to this. So I've got a demo script pulled up. When you are doing this kind of work in lab, what you want to do is have all of your plates ready to go. So be sample, couple wash solution, there'll be label reagents already in there. And then you'll simply take whatever protocol you want to start, press the start button, and you can add information like reagent lot information or sample ID and so forth. If you don't, just go ahead and tap the arrow to start it. And from here, it is literally just doing what the instrument is telling you what to do. You don't have to program locations or wonder where am I supposed to put the plates. So right now it's telling me to load the elution plate. It'll be a deep well plate. We just want to match the picture. There'll be a barcode in the back, and we're just going to load that onto the instrument. Press the arrow to acknowledge that we've done that, and it'll keep moving on. So now it's telling me wash plate 3. And then wash plate 2. And then the first wash plate. And finally, a sample plate. And then last, we're going to have our tip comb plate. This is, of course, what is going to cover our magnets. Once you get everything on there, it'll go ahead and start the processing. Not a lot of light in there, so I'll explain what's going on. But the first thing the instrument is going to do is basically pick up that tip comb. This is to make sure that it is attached. We don't want that magnet touching any of your solutions. So that has to be there. And there's a sensor for it. If it doesn't pass that, it will not continue with the process. This way, you'll never have to clean your magnet or anything like that. But when the tip is fully attached, it'll go over to the next step. You'll see that the magnet is not in the tip comb. It's just the empty tip comb moving up and down to do that mixing pattern, right? And once you're done with the mixing, the magnet will go back into the tip comb, and then it'll start dropping down through the solution so you get this even coating of magnetic beads. And you can have a number of those passes where you just keep piling those beads on to make sure that you are collecting everything. I've got 3 washes in here. They're going to be fast, medium, and slow, so you can see some differences in the speeds that we have that you're able to format. The last one will be the elution, which is at a bottom mix. It's also another different speed. The first one for the sample was a half speed. We use that in conjunction with fast for a lot of sample plate processing. All right, once you've collected your beads, we simply pull that up. That'll move over to the next plate will move underneath. That will drop down, the magnet comes out, we let go of the beads, and we basically start that process again with the mixing. Now, this is going to be a fast mix. We use this a lot for nucleic acid work, and it makes sense. The faster you mix, the more you'll get your impurities out, and so the better product you'll have at the end. Great for pretty much most nucleic acid work. And then the magnet goes back, we collect our beads so we can move them over to the next wash. That'll be a medium. It's half the speed of the fast, and this is where you get into the vast majority of your protein work, long RNA washes, things like that. So we're just slowing it down a little bit. All right, while we're collecting our imaginary beads, we have a nice touch screen that gives you a lot of information on what's going on. You've got 2 different countdown timers. There's going to be one to the end of the protocol. The one on the other side is to any comeback steps. Sometimes you'll have a kit where you'll have to come back to the instrument and add a reagent, like with our RNA protocols, where we have to add some stuff back after our DNA step. And that'll give you the time for when you need to do that. All right, we went over to our second wash. This is a medium speed. If you can't tell, it is a little bit slower. So again, most of your protein work and long RNA wash work and things like that for the process. Once you're done, again, magnet goes back, we pull up the beads, and we're going to move them over to our last wash, which is going to be a slow speed. That's more like mimicking a tube that's being turned over, okay? So sometimes you need those really slow speeds. For example, when working with whole cells, you don't want those shear forces to kill them. Sometimes protein work that has fragile kinetics binding to your antibody or tag, you might need to slow things down a little bit as well. So it does have its uses, but you'll see it's not a very vigorous movement. So we don't really use it for like washing and binding and elution and so forth. But it is an option on the instrument. All right, we're pulling out our beads in that medium wash. We're going to go over to our last wash, that's slow. We're going to drop our beads. Magnet comes out, we let go, and then we go through with the washing motions. So if you can't tell, that is actually pretty darn slow. You can see why we don't use it for actual processing. As that keeps going, we got a couple of other things on the touch screen. We have the total number of steps. So right now we're on 5 out of 8. With the name of the current step, which is a wash slow, and then you can also abort or pause protocols if you ever need to. But lots of information to tell where you are on the protocol that's being run. All right, so we're collecting beads out of that slow wash, and this is a typical process. You usually have sample plate, 3 washes, sometimes 4, and then an elution. So the 8-spot turntable is enough to accommodate all of that. With nucleic acid work, a lot of times you have to evaporate your residual ethanol from that last wash. So we incorporate a dry step. That is literally simply just letting the magnetic head hold on to the beads and hovering over a plate. So there's not much on there. Remember, we're not really picking up liquids. So it only takes about 2 minutes or so to evaporate that off. From there, we're going to go to our elution plate. Now, this is the bottom mix. It's very vigorous. At this point, we just want to shake the heck out of those beads because we want to knock that nucleic acid or target off. So you'll see it's just a mixing pattern at the very bottom of the plate. You can't see it moving up and down a whole lot because it's just almost like a vibration at the bottom over there. You can have a heat block that comes up. That'll be in the base of the unit. So you can actually heat or cool any one of these spots, heating up to 110 and cooling down to 4 degrees, which is nice because if you can't come back to the unit right away, then you can store it at that temperature for a little bit. But this is pretty much what a process looks like. And once we've collected all of our beads from our last plate, we're just going to take that in the tip comb, drop it, and that'll finish out the protocol overall. So from there, we'll just let that go ahead and drop. It's going to just have you unload the plates. And there it goes. And that's simply just a matter of taking everything off of the instrument. And all right, one more to go. And once you take that last one off, that'll finish out the protocol. The report is automatically created. It's in the instrument where you can download it if you'd like, and then basically start another run. So a lot of the nucleic acid protocols, they are kit-based, which means you just follow the instructions. There's validated scripts. It doesn't take very much to get started, and you can see it's easy for the operation. All right, let's move on to the next section then. So I just want to kind of introduce some nucleic acid kit options real quick. Again, not a deep dive. This is more for awareness. But before we do that, in the big picture, this being just the magnetic bead mover, we actually cover a lot of different applications. We generally divide them out into 2 different categories. There's going to be your nucleic acid and then your protein work. With the nucleic acid, DNA, RNA, total nucleic acid, we can do PCR cleanup, we can do plasmid mini preps. I'm not going to talk about all of those kits because we have over about 20 of them, but I'll highlight a few that might be of interest. Those come with validated scripts, but remember this has software to do script writing and editing, and you can always take those scripts and make changes or customizations to them if you have a unique sample type that needs a different workflow. On the bottom side, we also have protein work. It's still magnetic bead based, but what we do is we put a tag or an antibody on that bead and we fish out a protein target of interest. When we think about it in that perspective, that's your immunoprecipitations or IPs, your co-IPs, different types of protein and antibody purification. Phage display is a screening application. We can do protein digest and peptide cleanup before mass spec analysis. And we can even isolate larger things like exosomes and whole cells. They express certain molecules on the outer surface like CD molecules, and we can actually target those for enrichment or depletion. Now, these are not kits. They're usually just beads. Proteins vary quite a bit from each other, so workflows tend to differ. But this is where the software becomes super beneficial because we can create those custom scripts to match the workflow that you actually need. But that's the Kingfisher in the big picture and all the different applications that we can cover in more general terms. In terms of sample types, again, we pretty much cover all of it, whether it's blood, tissue, swabs, cells, urine. Really, what you're doing is just finding the kit to match your sample type, and of course, match what you want to isolate, whether it's DNA, RNA, and so forth. And different category groups like food safety, animal health, we can work with things like stool samples and so forth. Again, it's just finding that right chemistry. Most of the protocols, as you saw, are pretty streamlined. There might be an onboard lysis, but outside of that, it's just doing the binding, the washing, and the elution. The majority of protocols take under an hour. Some can take as little as 20 minutes to get it done. All right, just to generalize on some kits that you might be interested in, we have the MagMax DNA Ultra 2.0 kit. This is a general purpose DNA kit. And so what we do is we can isolate two different types of samples, your biofluids, which are going to be things like your blood and saliva and so forth, and then your solids, which are your cells and your tissues. There's going to be maximum limits. We don't want to overload the system, obviously. We can talk about what those look like, but this is basically the kit if you just want DNA from those samples. As an extension of this, last year we added what we call a booster solution, just a special liquid we added to the process that stabilizes those longer fragments. You know, these days when people are doing sequencing, they might be doing long read sequencing like PacBio and so forth, right? And so by adding the booster solution, you know, the kit already does a good job of getting fragments up to 30, 40, 50 kb. But now we can stabilize those longer fragments into a few 100,000 base pairs and have that available for sequencing, your long read sequencing purposes. As sort of a different extension of that, we have our sequential DNA RNA kit. So sometimes your sample is very precious. And if you want DNA and RNA from that sample, you don't really want to split it because right away you lose 50% of your sample, right? And so the sequential kit, right now it's only validated for blood and bone marrow. We're going to look to add tissues and cells to it, but it's basically where you process to get your DNA. Your first plate is going to have your RNA in it. So then we just add some beads and buffer, and then we're going to run it through a second protocol to get the RNA. So you get those in two separate eluates, but you never had to split your sample. And then in a different direction, we have a multi-omics kit. So these days, a lot of people are looking at different things within a sample to get a bigger picture, right? We can look at DNA to look at the genomics, RNA to get transcriptomics, proteins for proteomics, and of course metabolites as well. But sometimes people, again, have precious samples. You don't want to split. You want to get as much information from that as possible. And so our sequential protein DNA RNA extraction kit is our answer to this. Basically, you'll extract the proteins and get them out of solution first. Then you'll go into a DNA protocol and then an RNA protocol. So you'll literally get all three of those things from the same sample. Really nice option to be able to automate this on the Kingfisher. We also have a general purpose RNA kit. This is the MagMax Nirvana Total RNA. So it is total RNA, meaning you will get your microRNAs from this as well. But just like our DNA kit, it's a general purpose. So we split it out into biofluids and cells and tissues. In fact, just to get you an idea of what these workflows are like, basically like 5 different workflows. The cells and tissues all run on the same script. Their workflows only differ in terms of homogenization of tissue and when you add your isopropanol in the process, but they'll run on the same script on the instrument. And then for biofluids, we have our blood, serum, plasma, urine. For blood and serum and plasma, those just have a proteinase K treatment up front, and then they'll be run on the same script as well. And urine is just a little bit different in terms of how we do that upfront lysis, but it will also use the same biofluid script too. So lots of different applications and sample types that we can cover with the MagMax Nirvana kit. I also understand y'all are doing a lot of plants over here. So plants are a little more challenging. You know, if you talk about a DNA kit, DNA is DNA. Sample types might need to treat them a little bit differently. But from a single plant, right, you got leaves, you got roots, you've got seeds. all those different parts have different characteristics. And then, of course, you have tens of thousands of different types of plants around the world. It's really hard to get one kit to do everything. But we do have that one kit for both the DNA and the RNA. So it's the MagMax plant DNA, MagMax plant RNA. Just remember, because of the different characteristics of plants, the amount of polysaccharides and polyphenols that they have in there, whether it's soft tissue like leaves or woody like bark, there may be some modifications likely needed to that process, but we can help out with that as well. And then lastly, just real quick, we have some pathogen kits. Won't go into a lot of detail, but this is our first generation MagMax viral pathogen. The first kit is for gram-negative bacteria and virus. We can add an enzyme to that to get our ultra kit. That's for your gram-negative bacteria, your virus, but also gram-positive, your yeast and the fungus. And then instead of an enzyme mix, we have our microbiome kit. And what we do is we pair that with bead beating tubes in a different lysis buffer. So now we can get our dirty samples. These are things like your fecal matter, soil samples. I kind of lump insects and stuff like that into that kit as well. But this is a nice solution for it. Well, about 2 years ago or so, we came out with our second generation series. Both kits are really fine. But this one, if you haven't started, I would gravitate towards this just because it is improved chemistry, better pH stability, longer shelf life, and even a little bit better results. But it's still the same concept. You have a base kit and you either use it by itself for the gram-negative bacteria or virus. You add an enzyme mix to then also get gram-positive yeast and fungus. And then instead of that, you're beading and lysis buffer for your fecal matter. All right, so those are just nucleic acid kits I wanted to highlight. Again, we also have some additional ones. If you have any questions on what else we might have, feel free to ask on those. For protein applications, we'll keep it nice and easy. We have 2 different platforms for that. There's going to be Dynabeads and our Pierce beads. For the Dynabeads, they come in a variety of sizes from our M450s that refer to it being about 4.5 micrometers, all the way down to our Dynagreen, which are our environmentally friendly beads, really small size, so high binding capacity, but they all have different purposes, whether you want to isolate cells or exosomes, proteins, or doing peptide cleanup and things like that. Overall, the Dynabeads do have a lot of different types of applications. If you're looking for the best purity, they are actually the gold standard. But you can see all the different areas with nucleic acid and protein isolation, target enrichment, T-cell activation, immunoassays. We pretty much cover all of it with those Dynabeads. And then for our Pierce beads, same thing. They just tend to be a little bit larger in size, and they also have a Poisson distribution for that size. They have a little bit higher binding capacity as well. So these are usually involved more in protein and antibody purification. But we have protein A, protein G, and we have a lot of these similar ones for Dynabeads too. But this is where you'll find a lot of the tags, like your flag tag, hemagglutinin, that we have streptavidin beads and so forth. With the Pierce beads, we generally divide it up into 2 different ways. If you want to do recombinant protein purification, it's really yield versus purity. And if you follow the flow chart, we kind of help you make those bead decisions ultimately with the type of bead type that fits your workflow. And then if you want to do antibody-based purification, this is more along the lines of scale. Again, I'll just keep it really brief. You would just follow the workflow, whether you want to do large scale or small scale, and then you need protein A, protein G, protein L, streptavidin beads, and so forth. So we have a lot of different types of products for this. I know that's kind of hard to navigate, but we have good resources from us to help you make those decisions. So that's pretty much it. Just kind of wrap it up to say we have a lot of support with our tech support, our field reps, TSS. We also have people like me that are field scientists, and we have access to all of our R&D groups too. We do method development, customization. We can help with our products on that end. Just really great customer service overall to get you what you need. So with that, I'll close it out and I'll take any questions. That that is going to be our sequential. It does the protein first, then the DNA, and then the RNA. So it will work with a variety of sample types, tissue included. It's kind of our answer to the QIAGEN All Prep that's been around on the market, except they do DNA, RNA, protein. We do protein, then DNA, then RNA.
So I have a little bit of plan people. You know, in fact, I thought you have the plan management meeting and so it's not, I guess it's a similar workflow to everything else or,
It is, yeah. Now there's going to be some offline processing because you have to homogenize your tissue and so forth. This is not a beater, grinder, or anything like that. But outside of that, yeah, it's still the same workflow where you bind, wash, and elute. It's just going to be different set of chemistry, maybe different number of steps and so forth. Yeah. But a lot of times those kits do require modification. It might be the addition of things like DTT or PVP to mop up the polysaccharides and so forth. But we have a lot of tips and tricks on that. There is one, and it's very rarely used, so I'll have to look up the details. You may want to check back with me later on that. Yeah, that's kind of hard to do because I know a lot of times when you're processing like tissue with pathogens in there, you really just want the pathogen material and not the host. So it does get challenging to separate that out. I'll get your information afterwards and I'll follow up with you on it. Anything else? No, that's only one kit for the protein, DNA, and RNA. It's our multi-omic sequential kit. Mhm. Are you talking about just protein isolation? Mhm. Mhm. Are you wanting to maintain the 3D structure of that protein or? Yeah, you will get total protein off of that. So that'll be part of that first eluate. I mean, from there, if you want, you can do additional isolations and IP from that sample and so forth, but everything should be intact in there. Yes, that is available.
Good talk. I noticed, so first question, does the Kingfisher Flex and the Kingfisher Apex use the same plastics?
They do. Now, there is going to be a difference in that the Flex doesn't care about any scanning and barcoding, whereas the Apex does. So we manufacture all these plastics, take a subset, slap a barcode on it, and that's what's going to be used for the Apex. But technically, it's the same plastics for both units.
Okay, I noticed on the slides that the Kingfisher Flex can go down to 20 microliters. And then the Apex only goes down to 50. Is that just like,
It's just based on the heads. Yeah, it's sort of a generalized format. It really depends on the head format. They both have PCR heads, which work in PCR plates, and those can actually go down to 10 microliters on both of the instruments. The 96 deep well format, our speculo is about 50 microliters, but depending on what you're working with, I've got some tricks to get it down even lower sometimes. And then we have a 24 format, which is 5 times the amount of volume as the 96. That one gets down to somewhere between 30 and 50 microliters, but probably not much lower than that.
thank you.
Any other questions?
All right. Great. I appreciate all of your time. Thank you.
Just check real quick on Zoom. I don't see any on Zoom, but great. Okay. Thanks, Christian. Appreciate it. It's like, I think we are ahead of schedule here. Right. Hey, Lenny. Just thinking I went long. Oh, we're. All right. Well, good news, everybody. We are ahead of schedule. So, Susan has the lucky job of being what separates you and a delicious Qdoba lunch. So, always the enviable position. Susan Hetzel, she's a scientist with 12 years experience as a researcher at University of Minnesota in Minneapolis, studying the connection between biology and childhood psychosocial development. For 2 years at Thermo Fisher, she's been a field application scientist supporting qPCR as well as digital PCR and sample prep, focused on helping other scientists be successful. And so far, she's assisted the BioGarage with the Constudio 7 Pro qPCR and Absolute digital PCR platform. And I can certainly assist on a personal note that she's incredibly helpful. If you run into qPCR problems or something's not working, you need help with primers probes. She's a quick email away and easy to work with. Yeah, day or night, call her anytime. Weekends, just kidding. Oh, fun. Oh, cool. Microphone wireless.
Did anybody else get out yesterday and enjoy the weather? Because it was amazing. It was beyond amazing. I loved it. I went out by the Union and just watched the boats go by, watched rowing practice. Fun fact about me, since I'm getting this booted, I was actually on the rowing team when I was in college. Interesting fact. So got to relive some of my own college days. All right, so my talk today is going to be a little more centered around more traditional applications with gene expression and what we can do with RNA after we put it through Kingfisher extraction and what to do about that. So I have 1,001 windows open, because don't we all? That is how my mind works, unfortunately. Okay. I made my first QR code. I'm really proud of myself. All right. So today we're just going to talk a little bit about, I decided to focus on what most people do in the world of qPCR, which is gene expression, and sort of how to set the stage for creating a really good, solid experiment. So that's what we're going to do. I've got a 6-step process, and we're going to walk through each one of those and sort of talk about considerations along the way. I'm going to talk about this within the framework of qPCR mostly because that is more common, but I want you to understand it's not just a common name. We've got qPCR and we've got digital PCR, but that both of them use very similar reagents. So if you develop an assay on the qPCR platform, but you decide you need higher sensitivity, more granular details, you want to interrogate a slightly different aspect to that study, you can port those reagents and that assay directly over to the digital system, and you're off and running. Validation, I actually recommend happen on the qPCR platform or the digital instrument. It's faster, it's cheaper, and sometimes it's easier. You know, you've got 45-minute run versus hour-and-a-half run. Well, that's faster. So why not take advantage of that? So they really do go back and forth, and a lot of this will apply in both directions. All right, so essentially, we've got a qPCR study. We wanted to relative quantification. We've got a control group. We've got a couple treated groups, and we want to see how that changes with respect to these different sample types. Don't be confused. Relative quantification, gene expression, delta delta CT, they're all different words to do the same thing. Delta delta CT is the kind of math we're doing to get a relative quantification number. And we often interchange relative quantification with gene expression. So here's sort of the mental workflow we want to go through. Define what you want to do, find an assay and controls, verify everything's working, run your experiments, and analyze the data. We're going to focus heavily inside this red box on those 2 squares for choosing, obtaining samples and assays, and verifying that they're working. I trust that once everything's working, you can run the experiments, but how do we get this set up so the data we acquire represents what we're trying to interrogate? So, first step is obtaining quality total RNA. And Christian gave us a great talk about how to use the Kingfisher system, but what are some of these other methods? So organic extraction, I'm sure everybody's here has seen it. Somebody in the lab wanted you to do it. It's messy, it's stinky, and it's kind of scary. It's organics. Nobody wants to do that, honestly. Who wants to sit in the hood all day? We've got spin columns. Spin columns have their uses. They're great. Feeding tubes into the centrifuge is not always my favorite thing to do. And I don't know what sort of physical block I have with working with spin columns, but if you ever want to see a spin column fail, bring me in. I'm really good at that. So I really don't understand why, but here we are. So when I learned about magnetic particles, I jumped on board. So Christian showed these slides, but one thing I want to point out is even if you don't have the Kingfisher system, you can still take advantage of using those magnetic particles. You just need that separate stand magnet. And what you're going to do is you're going to move solutions around in front of that magnet, and you're going to hold on to what you want to against that magnet on your magnetic particles. Whereas in the Kingfisher system, we're moving the particles into different solutions. So if you don't want a pipette, use the Kingfisher. And everything's customizable. You can also mix and match. You can do some organic extraction and follow it up with magnetic isolation. It's up to you. But if you're hesitant to get started here, you can try those manual methods, assuming you have a standalone magnet. And if you want a little more assistance, I'm more of your local support, whereas Christian has this broad US territory. So I can walk you through how to do this, help you get protocols transferred, help you work on, I didn't get any material at the end, what happened? So I can help you with those things as well. We're both here for support. Like we said, our support second to none. So I love being part of this team. All right, so once we have collected our samples, how do we assess that we actually have good samples? So first, the easy one is taking an A260 280 or an A260 230. Both of those are good assessments of purity for different reasons. We want to make sure we actually have sample integrity. What do I mean by integrity? How chopped up is your sample? Do you have full-length mRNA? Do you have long strings, or do you have fragmented 50mers? If you have fragmented 50mers, you're likely going to struggle with your downstream applications and quantifications. You want to assess your genomic DNA concentration if possible. You can also do a DNA treatment, so you can digest that along the way. If you're going to collect DNA, you can do an RNase digestion. You want to try to eliminate contaminants as much as possible and make sure you have a good yield. So how can we do that? I mentioned the 260, 280. 260, 280 of 2.0 is great. And as we increase our protein concentration, we start to drop off. And that's where those numbers come from. That's why we actually look at it and what we're trying to assess. So 260, 230 is going to look more at those organic and other molecule contributions to fluorescence. So just keep in mind what you're going for and what you're looking at, but does take quite a bit of protein contribution to shift that. So if you don't have a 260, 280 of close to 2, you really want to assess your purification methods. All right, RIN score. So Dustin pointed out that the BioGarage has a tape station and a bioanalyzer. That's awesome. When you run those, what kind of data are you going to get? What we're going to get, they produce these simulated gel images. So they're not actual gels, they're computer-generated gels. And what we can do is sort of look down this vertical axis here at fluorescence. So if we're looking here, these are represented in fluorescence. So this is human. And one of the things we can see are these large peaks from our rRNA. We have 18S rRNA and 28S. And those should be in particular proportions when you have fully intact RNA. As that proportion shifts, you get a more of a smear of more smaller sizes. And we've actually, in this case, lost our 28S fragment entirely. And I also want to point out the scale here. We've got a lot of really great material here, and we lost it. So when you have a RIN score, that's what we're looking at, is the proportions of those 28S, 18S. And different types of samples, you're going to look at different peaks, right? So make sure that you're actually looking for the right thing in your sample, because this 18, 28S are not going to be in your plant samples. So when we're actually looking, when you actually run this experiment and you generate a number, you're looking for a RIN score of, for qPCR, you want it to be at least 7. Okay, that's kind of a minimum. Once you get below that, you're starting to get so fragmented that the chances of actually having what your analyte of interest be intact actually starts to go down. So now you're inputting this RNA, but it's not necessarily representative of what's happening in the sample anymore. So this is where you start to need to be cautious. So if you have RIN scores of 2 or 3, that's going to be a rough one. That's going to be a rough endpoint. And you sort of have to put a caveat on any of the data you generate that it might not be accurate. And unfortunately, one of the problems there is FFPEs. Really quite often heavily fragmented because of what has happened to that sample along the way to actually make it an FFPE sample. So be very cautious in those ranges. Here's some kits for RNA extraction. These are some of my favorites, but I am going to point out, I sent this separately to Dustin and some other people. This will be a link when it goes out, and that is actually a little search tool that you can simply say, this is what I'm doing. Here's what kind of system I want to look at. And I just check a couple boxes on the left and it'll give you a really short list. So you don't have to think too hard about which kit am I doing and how do I fit it all together? And if I use the prime kit, you know, which 4 components do I actually need? It filters it all down to a link that you can actually go and get a protocol and put it on the instrument. Okay. All right. So that's talking about getting good high quality RNA. Next, we're going to talk about reverse transcription. So reverse transcription is pretty important because RNA itself isn't going to amplify in our PCR style reactions. We have to convert it into DNA so we can actually do something with it and measure it. So how we do that is pretty important. Again, I threw in some of my favorites. The superscript Velo is our current favorite. HiCap is also a really good choice. Some caveats about timing and how that reaction is actually completed. So take these into consideration. But once you choose that, what do we need to do in the lab? And we actually need to verify that we get this one-to-one conversion of RNA to DNA. All right. How do we do that? It's a serial dilution of RNA. So before transcription, you take your RNA, you dilute it a few times, you put each of those into the same cDNA reaction or the same RT reaction to generate cDNA. And then we run that product in our qPCR reaction and we see how much we made. It needs to be linear. As you linearly decrease your RNA, your cDNA product needs to also decrease linearly. If it does not, you're outside of the capacity of the kit, right? That is your hard stop limit, all right? So in this instance, we can operate inside the yellow box, but not outside. And likely what has happened is we've put in too much RNA into that conversion reaction, and we're not appropriately accounting for it at the back end, all right? So whichever RT kit you use, read the directions, stay within the bounds, and then when you do, measure it. Also, when you're starting and generating this process, what we're talking about is process steps. This is not something you need to do for every single sample, but it needs to happen for your process. So once you've decided, these are my ranges, this is how my extraction works, this is how much sample I'm going to put in, you don't really need to do it again because your samples up front shouldn't be changing all that much. So this is a one-time kind of a thing. Check it every once in a while. I always recommend that. Go back to it. Try it again. All right. So once you have chosen and verified your reverse transcription, next you need to decide how you're going to run your qPCR reactions. And what I mean by that is, are you going to do a 2-step or a 1-step reaction? So a 2-step reaction is something where you're essentially going to create a cDNA library that you can come back to over time, use that library, run a bunch of experiments. That's 2 steps. You do 1 step for RT, and then you do 1 step for qPCR. 1 step is where you take RNA, you put it into a master mix with 2 enzymes, and you do both of those reverse transcription and amplification steps end to end in one tube. If you do that, you cannot collect cDNA and you cannot come back to it. Why would you ever want to do that? Because you now have an exact mapping match and you have fewer steps, fewer reactions, and fewer pipetting steps. So if you have an abundance of target that you don't need to come back to, 1 step is great. It's a great option because you're reducing your variability, but it does come with downsides. Science is great, but it always comes with trade-offs, right? You have to know what you're doing and you have to make a choice. All right, so once we've decided if we're going to do a 2-step or a 1-step, then we can start focusing on what kind of actual qPCR reaction are we going to do. The chemistry for qPCR comes in 2 main categories, CyberGreen and TACMan. let's just cut to the chase. If you're going to ask me which one's better, I'm going to pick TACMan. That's not just because I work here. That's because I was in your shoes for 12 years, okay? We'll get there, but the instruments in the lab, Dustin's instruments at the BioGarage, will handle both types of chemistry. They're all calibrated to do all of those things. So let's talk about why we would want to pick either one. Cyber Green is super fast to get going with. You need 2 primers and some dye, and you're done. Okay. The Cyber dye is a double-stranded binding dye. So whatever that is double-stranded in that reaction will start to fluoresce as you make it. You make more product, you get more fluorescence. It's great. What could go wrong? The problem comes in that anything that's double-stranded is going to produce fluorescence. So if you have primer dimers, it's going to fluoresce. If you have mispriming events, it's going to fluoresce. And that can confound and produce inaccurate results in that cyber reaction. Another point to bring up is homologs. Lots of these systems have known homologs. That is a part of evolution. You know, there's really no way around it. And the more homologs you have, the more issues you may have with accurate quantification in cyber. All right, so what's an alternative? We can do TACMan. The big difference between cyber and TACMan, we still have 2 primers, just like we did with cyber. We add a 3rd molecule, and we call that a probe. So in this instance, we don't have a non-specific binder, double-stranded binding dye. We have a specific reporter molecule, and that is the probe. So as we go through amplification, the probe binds, it gets cleaved during the amplification, and that is where probe signal, I hate the dentures, right? Aren't they terrible? They're awful. Okay. I should just turn it back to the old Pac-Man mouth. Anyway, we all run out of time sometimes. Okay, so we get degradation of the probe, and that is where our signal comes from. So we can have a lot of things happen in this amplification reaction that don't actually impact quantification results and endpoint and the ultimate fluorescence that we actually look at. So it's a safeguard. So we've got the same kind of amplification with cyber with this extra caveat that it's got to be this also before we get results. All right, so specifically, how do we avoid homologs with these TACMan probes? And really the special thing that happens is the enzyme has the ability, if there's something in its way, if it's weakly bound, the enzyme will actually kick that probe off. So if it's just a horrible mismatch, it's just not even in the way. It just kicks it off. If it's tightly bound, that it has to have an appropriate energy to that double-stranded binding. And if it does, it will come in and actually cleave it. There's a little arm on the enzyme that actually come in and cleave that probe. So if we have any even single base mismatches, that is enough destabilization in the probe that this enzyme will tunnel under and kick off the probe. So that's the real cool part of this. So I used to do a lot of SNP genotyping, 1 base pair mismatches. I struggled for years to create really good assays. To do that, I switched to TACMan. It was all over, never going back. It works so well. Also, another point to bring up about our TACMan assays is they're all designed to work at 1 cycling temperature. No matter what you order out of the catalog, it always works at what we call universal cycling conditions, which means you could go in that catalog, buy 6 different assays, throw them on 1 plate side by side, and they'll all work at the same time. I love that. All right, so obviously, like I said, I made the switch to TACMan assays before I was ever part of Thermo. So obviously that's going to be my choice. But all the instruments we have, qPCR and digital PCR, have the capability of working with both assays. We just want to make sure we put in good assays at the beginning to make sure our results are quality at the end. Once we've picked our chemistry, we need to optimize it. So if we're looking at optimizing a cyber green reaction, we have some considerations. How much primer, what's our annealing temperature, and caveats like that. And one of the tools we can use to assess quality besides sequencing, that's sort of the gold standard, did I make what I intended to make, is looking at a melt curve. The melt curve has some different features that can give you an idea of what's going on inside your reaction. So at these lower temperatures, we see the melting of maybe primer dimers that are in the reaction, which is normal but not advantageous. So it's good to know that's what it is. But we also can see these features like this, which is a non-specific product. Those can be really sneaky, because if that product were just a little bit longer, it might actually hide under our main peak. So just because you run a cyber melt and you get one peak doesn't always mean you made one product. Just kind of want to throw that out there and scare you into using TACMAN assays. All right, so once we've looked at those, that melt curve, and we sort of have an idea, we did our sequencing, we looked at our melt curve, everything looks pretty good. What's the next consideration? Obviously, you need to work at a temperature where you have good, strong amplification without a lot of those satellite products. I have gone down that rabbit hole and spent a long time trying to tease out the individual, tenth of degrees to get my reactions working as good as possible. That is a rabbit hole you go down and you'll spend a lot of time in there. Okay. After that, we want to check that we're using the right concentration of primers. So if you titrate down at, like we see in this example, we used 500, 300, and 100 nanomolar primer, excuse me. And what you're looking for is that we have good, strong product without a diminishing without a CT shift. So we're not seeing a difference in assay performance, but that we meet this full endpoint signal, right? So just double check that. If we look at gene expression assays, TACMAN assays, I checked, we have 2.8 million pre-designed gene expression assays. We have, I think it's over 20 million pre-designed assays in total. So it's huge. 32 species. We just updated our website, our search tool and our assay design tool. So I'm going to go over that really quick. There was a request to look at that. So if you go to this search tool page, the link is up there at the top. But really, what we see is we have all these different kinds of assays. You don't need to make the selection first. You can make it later. And then you put in a keyword. It's that simple. I want to look at beta-actin. I type in beta-actin, I hit search, and it will give me a list. You can filter that list for species, for assay makeup, transcript number, ref-seq ID, all kinds of things you can customize. So that is found here in this pre-designed, order the pre-designed assays. What do you do if what you need is special and not in the pre-designed group? Then we go to this order custom here. And when we do that, it sends you into this assay design hub. And from here, we have a forking decision tree. You can either request redesign services. So these are all submit a form, an actual human looks at that form and the wording of your request and makes a decision and makes a recommendation, does a redesign. Okay, so that's more for multiplexing services, or you've gone through some of these other tools and it didn't work. But these tools that are available for you, this assay design tool, the custom primers and probes, and even the digital custom assays are all available to you. And if you link into there, what you see is this list. This list has all kinds of individual tools. And each one of these you can drill into and say, I have a reference sequence, make me an assay. And it will generate an assay that you can order and get within a few days. Okay. We've got SNP genotyping assays, copy number variation assays, small and microRNA assays. And then here at the bottom, we've redesigned our tool here, which is why I'm going through it with you. I usually don't do this, but at the bottom here is this custom TACFan primers and probes. So we are redesigning this tool in our website to be more competitive with IDT. So you can come in and just type away and say, I want these primers, I want this amount, send them to me as fast as possible. Our turnaround times are faster than they've ever been. Couple of days for any of the primers, probes, obviously, as the complexity goes up, your time goes up. But, you know, FAM assays, typically you can get them again in a few days. Purification, again, we're always gonna take a little bit more time for purification. And so does IDT. But I've had a lot of people really rave about the quality that comes out of this. So all of these are available. Each one of these tools is gonna look a little bit different, feel a little bit different. Let's see, I'm gonna leave this really quick and show you what that custom ordering screen actually looks like. And I'm going to zoom in so we can actually see it. So the first step is just giving the order a name and then telling it what you're ordering. So this is a probe, this is a primer, here are my sequences. I'm going to give it a dye name, a quantity, a quencher, all of the typical things. And then it's going to give you a price. So as you have more add-ons, the price goes up. But, and we are going to require, probes are always going to go through HPLC purification. Always. For primers, you can have them sent dry. You can also have them sent to you simply desalted. Okay. So again, it should feel a little more like your IDT ordering page, but have a little more security that you're actually ordering from Thermo Fisher. All right. So just wanted to highlight that. And once you put it in here, it's going to review it to make sure you don't have any horrible problems. And then obviously we're going to give you the option to add more reagents, master mix, etc. And then you can order. We have other options. If what you need is not here, we can offer mixed bases. We can offer lactic acid. We can offer a lot of those crazy things. We just don't put it on the website because that's not the high throughput, fast stuff. So if there's things that you need, I'm continuously surprised that we offer that. Yes, we do. We offer that. So let me know, and we can work with our specialty team to see what it's going to cost, how long it's going to take, and make sure we actually fit your needs. Okay, that's enough of the sales part of this. Let's get back to some science. All right, so we picked an assay. That is fantastic. Life is wonderful. All right, so once we've chosen and optimized those assays, we need to go to the bench. We need to test some things out. And some of the things we haven't talked about yet are your endogenous control gene or your normalizer, assay efficiency, and any of that genomic DNA contamination. So there's, again, this is one of those terms that has a lot of duplication in qPCR. We have a normalizer gene. It can also go by endogenous control gene, housekeeper. Those are all normal words around your controls. Key points that need to be considered is that your endogenous control or your normalizer genes needs to be consistently expressed in all your sample types. How do you know that? You test it, okay? If your control gene varies in your sample, in your treatment conditions, in your tissue types, it is not an appropriate control gene. Candidates, common candidates are 18S, beta-actin, GAPDH. But that doesn't mean because somebody else used a control gene that it's appropriate for your experiment. It is something that has to be tested, and you are responsible for that. It's something that kind of happens now with the pressure to publish is greater than it's ever been, and it's kind of overwhelming. But you have to remember you are putting your name on this and your reputation behind it. And if somebody else made a mistake, yes, it's their mistake, but it's all your mistake to follow their mistake. Okay, you've got to check it. And that is an experiment that needs to be done. It doesn't need to be an exhaustive experiment. You just need to check it. All right. So how do we check it? An example would be to take a few treated, a few untreated samples, a few different tissue types, whatever the variables are in your experiment, and do your reverse transcription, run those samples for your endogenous control. And then this is a really fun plot that the QuantStudio 7 Pro will actually output. And it sort of gives you that visual of, is it a good endogenous control? So we had 4 candidates here. 3 of the lines are super flat and one of them's not. So I'm going to say the one that is not flat is not a good endogenous control. It's kind of that simple. The software will put some numbers to what it thinks a good endogenous control is going to look like and give you the best candidate endogenous control gene for your experiment. But it's always safe, especially when you're starting something new, to try a few and see which one works. You can have, 3 or 4 of these in the freezer and try them out and see which one's actually going to work. All right. So the next thing is to verify the efficiency. And this is really important, especially when we're in the world and talking about cyber. If you have an efficiency that is well below or well above 100%. So what are we talking about? We're talking about at every cycle, do we get 100% growth? Do we see a full 2-fold growth? Okay, remember, we've got 2 primers. We should have twice as much at every cycle. That is what this number 2 here actually represents, is that 2-fold growth. If at every cycle you're only seeing 80% of that 2-fold growth, that equation is now not correct, okay? Which means your results are not correct. So we can tackle this in 2 ways. We can either get a better reaction, get better reaction conditions, get that better to 100% efficiency, or we can change the math. The software will allow you to change that math. I prefer seeing 100% efficiency because that's a good indicator that you actually have a good assay. If you have 80% efficiency, it's a good chance there's something going on that you're not addressing. So how do we actually do that? What we do is we take our assay and a few samples, 1 sample, and run a serial dilution. We run that serial dilution and then we look at the slope of that line. If we have a slope of 3.3, that is the magic number. 3.3 means that we have 100% efficiency based on a 2-fold growth process. Then we're good to go. If we don't, again, we can either address our assay or address our math. Okay? I'd prefer you address your assay. Right. Questions about this? I kind of flew over it and it's a pretty important point. Just run a serial dilution. Is it linear? Give yourself some grace, 90 to 110%, great range to be in. Everybody's got pipetting issues, which is why we give ourselves a little wiggle room. But if you're much outside of that, I would really go back, take a step back and ask yourself why. Is this really what I want to do? And run your controls. There's no substitute for running appropriate controls. Run your positive and negative RT step, run your cDNA and no cDNA controls. Just make sure that amplification and you get no amplification when no sample is present. There. Okay. In summary, make sure you have good RNA. Pick a good reverse transcription step and make sure it's working. Let's see. We've got pick your chemistry, pick a good test of that chemistry type, and wet bench test everything. Okay. I'm not going to ask for a show of hands, but I know all of you did this the last time you sat down in the lab to do a gene expression experiment. And the reality is people don't do this and they need to. Okay. We are all putting way too much faith. And I see this. I see this in papers. I see this from researchers. People are going and using web tools and not doing the testing. I've seen, reviewers not ask for this information. It then gets into the public space as validated and published, and people are reusing this. And I'm very, very scared that we're making some big missteps and building off of not a solid foundation. So this is my call to action for every one of you to take that next step and actually do this correctly. Okay, how much time do we have? Oh, all right. Who wants to hear about digital PCR? Yay, Dustin. What do I need to tell you, Dustin? Let's talk. Okay. I love digital PCR. It makes me so happy. Okay, I got sucked into this digital PCR world, and I've actually become one of the North American experts with the Absolute Q system. So here we are. I'm going to try to give you some application overviews, but I want to remind everyone that we can take everything we did in our previous qPCR talk and pull it straight over to the Absolute Q system. All of those assays, the verifications we did, we can put directly on the Absolute Q system. So if you have a study, I think I mentioned this already, you have a study and say your gene expression, your error bars just overlap just a little bit, and you want to put the p-value on it, but it's just not quite there. That you can put on the digital system because we have a different kind of data with a different kind of precision, and you can see, is that a real effect? What kind of p-value can I get out of it? So you can sort of see the in-between spaces. Okay? It's not just rare targets. It's not just low concentration. It's any small differences across the board. Okay, let's get the wheels turning. I know you guys all have data and you're like, oh, I wish, or I want to know. Bring that in because we can take a look at it. And because we can pull from what we learned from qPCR, we can get going. And we've got millions of TACMAN assays in our pre-made catalog that we can put to use on our Absolute Q system. So when do we pick and choose? Absolute Q versus qPCR. The qPCR is great for relative quantification. It's great for big differences and screening experiments. It's great for high throughput. You can get a lot done and it's pretty cheap. So in the lab, everything's a trade-off. And in this instance, we're going to trade off speed and cost for increase in precision. Okay, that's really it. Everything's a trade-off. You're not going to do a screening experiment on the Absolute Q. Too low a throughput and it's too expensive. But you want a reflex test on the Absolute Q to see the in-between spaces. Let's go. So what can we do? We can obviously do absolute quantification. How much contaminant do I have in this soil sample? Can be really low concentration, but we can quantify that very accurately. Copy number. So by this, I mean, how many copies do I have of something in the genome? Rare target detection, that is going to be falling into things like, classic example is cancer. You have 3 cells in a tissue sample that are mutated, and you need to know that those 3 actually exist and that there are actually 3. That is a great thing for the digital system. It's very, very accurate. We have patient sample data going down to 0.001% mutation in a sample, which is pretty incredible. It's so anything that's a needle in the haystack, that is a great digital application. Gene expression, I've mentioned this about 5 times now. You know, if you're looking at small changes, you can see that in the digital data that you maybe can't see from qPCR. The best systems from any vendor for gene expression in qPCR are going to have a limitation of about 1.5-fold change. Okay, that's a pretty good. But if we compare that to the digital system, we can see 10% changes in the digital system because our error bars get so small. We're collecting a different kind of data. Molecular integrity, I don't think that is of use. Drop-off assays, CRISPR genome editing. That is something that the BioGarage can help you with. And that is a good application here. So this is that classic qPCR versus digital PCR data. We made the same reaction. We put some of that reaction on our qPCR instrument. We put some of that reaction on our digital PCR instrument. These are direct maps 1-to-1. And you can see that our error bars are decreased to the point where we can accurately quantify those 10% differences. The qPCR data is not bad. This is sort of what's expected, but the digital's better. The other big trade-off here is that qPCR has something like a 9-log dynamic log, okay? The digital PCR instrument is going to be much smaller. You want to look at accurate changes, you're going to lose dynamic range. So you've got to have a narrower focus on concentration. When we compare the math between the two, we now do away with that 2-fold growth. Okay, because digital PCR is looking at data at the end of amplification. It's just the kind of data we get out of this. We don't need that 2-fold growth anymore. So the math looks a little more normal. I hate log math, exponential math. It's just not how my brain works. So we get back to more normal math. All right. I mentioned genome editing. So this is, do I have an insert? Do I not have an insert sort of question? And how often does that happen? The absolute Q system, the way this experiment was set up, we have forward and reverse primers around the cut site. We have one probe that is associated with the sequence within the primer region, but not part of the cut site. And then we have another probe over the cut site. So what we can see here is if we pull this out, the green actually is the, let me see, that's the uncut. And if we have red, that's the cut. So you can not, within a mixture of samples, look at how much cut versus uncut you have. Rather than just averages, we're looking at individual molecules. So I also want to point out in this instance, we use probes to do this. If you can reconceptualize this, you don't always know what you're getting when you do genome editing. It's kind of variable sometimes. It would be great if it was this predictable, but it's not always. You can employ a cyber reaction in this case, and maybe put one of the primers across your cut site and sort of expand the utility and make it more universal. And you don't have to wait for probe production. You can just, get that screening done faster. Going to be slightly less reliable, but again, trying to, design probes in this way would be expensive and time consuming. So if you're talking about screening, cyber might be a good application. And none of our materials actually say that you can put cyber on our absolute Q system. It's a marketing decision, not a science decision. So I typically don't recommend cyber for digital PCR applications. I like to pair the most sensitive technology with the most sensitive chemistry, which is TaqMan. But in some cases, there are uses. And I think this is one case where cyber might be a good solution. So it can do it, even though none of our literature says it can. All right. Liquid biopsy or tumor testing is another application that we like to put forward with the digital application. That really, again, we're talking about needle in a haystack. We're looking at circulating tumor cells, cell-free DNA, and what sort of changes do we see in that sample? So in this instance, we can also look at response to therapy. We can look at driver mutations, and we have many, many offerings within the catalog that has been sort of tuned for the digital application. And those are listed here. Don't take a picture of it. It's too big of a list. I'm sorry, it's not easy to read. I just want to point out, we spent a lot of time in getting this put together. If your mutation that you're interested in looking at is not on this list, that's great. It's fine. We have SNP assays for a lot of those SNPs in our general qPCR catalog. We've actually pulled everything together. It's not just one catalog. We don't have a qPCR catalog and a digital PCR catalog. We just have a TACNet catalog now. One thing I will point out is our fusion transcripts. If anyone's ever looked at BCR-ABL, we have not only specific assays for these, but we have it developed as a multiplex. So you can look at all of these mutations together from one sample and quantify them individual in one well. And with synthetic controls, we've gotten down to 0.001% mutation. That's really low. That's cool. So I didn't already say it. Here's a picture of the absolute Q system. This, I have personally done 4-plexes. Bamvik, Abby, and Sci5 are my favorites. We've got a new software upgrade. We can now do 5 colors, plus a 6th that is our QC channel. So improvements. It's easier to multiplex on the digital system than it is on the qPCR system. In qPCR, we're looking at the data we use for quantification. We're measuring during amplification. Okay. The data we use for quantification in the digital system is looking at the data when it's done with amplification. So we spent all of that time talking about assay efficiency and how it affects quantification. Moving into the digital platform, because we're looking at the endpoint, we're just looking at did it amplify or not. So it doesn't really matter how we got there. So efficiencies don't matter as much. So interactions with multiplexing, which can affect efficiency, are now no longer as a high priority. I won't say it's not a priority. It's just not nearly the high priority it is with qPCR. So I want you to think about what you've got going on in the lab and what you want to get done, and then start searching the catalog. Because again, your job is to get publications out and use the tools that you have available to get that data generated. Use the BioGarage, use our services through Thermo Fisher to help you find those assays. I don't want anyone going back to their lab thinking I need to start from scratch to get a new assay going, to get a new project going. Use your tools. Use me, use our technical support center. They're qualified scientists too. They're not just phone people. If you call them, you're actually going to get a scientist and they can help you make some of these decisions too. Or if you're just stuck trying to use the website, they're good for that. But I'm always around, like Dustin said. 24 hours a day, 7 days a week, right, Dustin? Thanks. Appreciate that. Thanks. So again, Wisconsin's mine. Nobody gets to have it. It's my territory. This is, I'm here. I am interested in making relationships and helping move your science forward. So if you want to start a new project with qPCR or another TACMan solution, copy number, SNP genotyping, you need to talk to someone about how to get this going. And Christian's busy. I can help with that too. You want to do digital? We'll do some digital. So, question.
I can at least think of one while we're waiting. So maybe I just can't think of it off the top of my head. But so you emphasize the importance of controls, which we all always do all the
Always.
time.
Wait.
Like, in the case of like one step qPCR, like how would you go about like what controls? Some options.
Yeah, so it's probably a good idea. Most of the one-step master mixes, those enzymes are available as a two-step. So if you feel a need to check that, you can. But the idea is that you still run that serial dilution with the RT and cDNA. So you do the one step in a serial dilution. If you feel like there is an issue, you can take a step back and run that as a two-step to check.
Any other questions about
Yeah.
Name of the machine you use for the digital PCR, and do they have it at the BioGarage?
Do. Yes, Justin has been an early adopter with the digital platform. He's been amazing. I love working with him. The name of the instrument is, if you want the full name, is the Thermo Fisher Quant Studio Absolute Q digital PCR system. We typically call it the Absolute Q. It comes with specific plastics that you need to use. I highlighted that the consumables overlap a lot with qPCR, but the plastics you do need to have, they are unique and available. Yeah.
Exactly. Yep. Instrument.
Yeah. And it's really user friendly. It's not like other, I don't want to talk bad about anybody else's instrument that's on the market, because essentially at the end of the day, we're all kind of doing the same thing. But as far as usability, somebody who's never run it before, being able to walk up and use it, I've had people actually just pull up the user guide, read it, and run the instrument and get data, get good usable quality data. It is possible. If you want somebody like Dustin or Molly to help walk you through it, they can do that too. They've been trained, they know what's going on, but it is honestly prepare just as you would the qPCR style reaction, where you have the appropriate digital PCR master mix with your assay components and your sample. 9 microliters goes onto the digital PCR. It's a plate. It's a typical plate size format. It goes into the system, and an hour and a half later, you have your data. Those are all the steps you need to do. It's really that simple. I wouldn't say that if it weren't true.
Yeah.
But I have typically have very high success with brand new users.
Yeah, so unlike other digital platforms, you don't like have to make droplets or worry about weird surfactants or any of that stuff.
It just
You
works.
just prep
It
it.
just
and
works.
That's where.
It's easy to get behind something when it just works.
And the software is actually really easy.
Yeah,
Compared-
and it's getting better with
intuitive.
every iteration.
Wow. Okay.
Yeah.
That's exciting.
I know. Yeah.
We're gonna do learning sync with them. But I'm pretty sure I'm gonna interact with you later after we get some results, because we're gonna have to validate. So you talked a little bit about FFP samples. Our rim's not that great. It was like 8.4. However, they said to us to do like a DV 200. That was great. There's another QC you can do. And I just don't know, like, any advice on it. Like, we will have to do some validations. We will work with you, and I will probably interact with you. Any advice on it? Like,
Besides just order from the catalog. That's the easiest one. Something I didn't bring up is that we have what we call our TACMAN guarantee. If you order a TACMAN assay and it doesn't perform the way we said it should perform, we give you your money back or we give you a replacement. That's the kind of confidence we have in that catalog. But so RNA-seq validation, go through when you pick your assay, make sure that the assay you're choosing is for your correct transcripts. With the RNA-seq data, you're going to have a lot of that. So make sure that the assay actually sits across an exon-exon junction. That's going to help with that genomic DNA contribution, it's going to automatically be removed from the assay. Yeah, it should be pretty straightforward. I don't have a lot of people struggling with that validation. So do you have a specific concern other than I
Well,
need to do
that's very
validation?
degraded. It's not good quality.
Yeah. In that case, I'm going to encourage you to find the smallest assay that you can, the smallest amplicon. So as you go up in size of your amplicon, you require that intact sequence to be that long or longer. So the smaller that amplicon is, likely the more accurate that qPCR quantification is going to be. So if you've got an average of 200, I think I heard you say that, hard to hear. And you go for an assay that's less than 100, an amplicon that's less than 100, you should be pretty accurate, but it's going to be based on that normal distribution of degradation.
Actually, 2 years ago, I have the one times with the demo of the PCR user. And I have a question. Is it for the Pac-Man, the primer? It's how long it can keep use? Is it expired?
Oh, how long? So one of our TACMAN assays, I think we put an expiration date of approximately 2 years on those. But quite honestly, in my hands, I've used them for a lot longer than that. I think it's sort of encouraged that we just put an expiration date on anything that comes out, but I haven't actually had one degrade on me. Have you? In my, like, I have to give you like the company line that says it's 2 years or it's 4 years or whatever it is written on the tube. But in practice, it's a lot longer than that.
Oh, okay. Because if you buy the company, because you have a website, they have a common primer use, and then your order, they have expired date or not. And then it's the 4 hour that have a special 4 hour primer gene. We want to keep that by hour. And then that's primer. We know how long can.
Right. Well, I mean, and honestly, oligo and primer probe stability is ultimately dependent on storage more than anything else. How many times did you pull it in and out of the freezer? What concentration was it at when you were in storage? Did you store it at -80 or -20? How did you handle it? I mean, there's a lot that goes into that, but I've put my assays through some pretty brutal handling and I haven't seen degradation. So I have to pull the company line. If we're going to have a guarantee behind this, we've got to have an expiration date on it. But the practicality is it's much beyond that. So if you have that, I would encourage you to try it and test it. If it doesn't work, get a new one. If it does work, continue on.
Actually, it's the primer so expensive. Yeah, it's a kit, it's okay, but each primer is very
Yes.
expensive.
All of
And
those are
then it's
expensive.
when you use your covered light, avoid.
Yes, please do.
Then also keep the R is -20. You said to keep the -20 when you use your R is to keep the ice melted.
Yeah, so typically, again, this is what's the company line versus what I actually do. When I was in the lab, like I said, I was pretty brutal on everything. I was always worried about photo bleaching, so keeping it away from light. Your fluorophores will degrade over time under standard office light. So I would encourage you to use foil or something else to keep it covered when you do setup. Keeping things on ice is always a good idea. Did I always do those things? No, I did not always do those things. Should I have always done those things? Absolutely. I should have always done those things. But the reality is our enzymes our TACMan enzymes and our master mix. Most of the modern ones, so anything fast advanced master mix or a digital PCR master mix, have something like 72-hour benchtop stability before enzyme activation. So we've got a lot of robustness against a lot of these lab processes. So, typical oligo usage, create a working solution that you can pull in and out of the fridge or in and out of the freezer on a regular basis at a reasonable concentration for benchtop use. And then you can keep an aliquot in -20 or -80 storage, and that's your safeguard. If you are keeping a solution in the -80, I would have no problem saying years and years, 4 or 5 years of stability is totally fine.
Very good. These things, like do qPCR, we need the triple. So for each sample, but this one's only one sample. It's so convinced.
Yeah,
Yeah.
always
so
run in
it's very-
triplicate. It's the smallest number you can do outlier elimination on. So did you do it right? Well, try it 3 times. If one of them's wonky, throw it away.
I might add just a couple of things. It took us a while to realize, I mean, you're right that the TACMan probes, that's not exactly free. It's fairly costly. It took us a while to realize that there actually isn't. Maybe it's only for like the cataloged items. There's like the small, like the micro, there's a smaller unit you can get that I think is like $100. You can still get like 100 reactions out of it, but
Yep.
if there's something you're not, if you're going to do tons and tons of reactions, you want bigger volume. It's
Right.
cheaper. But if you're just going to do a few, there is
Yeah.
cheaper volume.
It's the smallest size will provide you, like Dustin said, is about $100, and what you get is 75 20 microliter reactions. So you have the 3D 4-well block and can do 10 microliter reactions, which means you now have 150 reactions for about $100. So that is a very reasonable, let's give it a try kind of a cost. Especially if it means you don't have to do all that optimization you need to do with Cyber. You don't have to do the temperature gradient to determine your annealing temperature. You don't have to do all those melt curves and all the time you spend stressing about making the right product. Again, I was a researcher for a lot longer than I have been a part of Thermo Fisher. And one of the joys in my job is that I get to make scientific recommendations for you. And that is what I'm doing. I was a TACMan user and I believe in it. I'm going to recommend it to you because it really is a better choice. Wow, that is a bold statement. Okay.
Any other questions?
Just like for my edification, I guess. So you had mentioned for the digital PCR, like the efficiency isn't, say, as important compared to qPCR, but can you touch on that a little bit more? Because I would think if what matters is like the end quantity, that efficiency would affect that, but maybe I'm misunderstanding.
Thanks. Since I just closed my presentation, I have a perfect slide for that. We'll get it back up. But essentially, so we have the same kind of reaction between the two platforms, but how we gather the data is completely different. The first step in the digital system is that we take that bulk reaction mix and we force it into micro chambers. So every bulk sample, every reaction that we create, we push it into 400 picoliter size reaction chambers. And what we're forcing by doing that is getting individual molecules, target molecules, into individual microchambers. So I'm a very visual person. I like visual examples. If you go to the midway, imagine we're at the fair. Okay, we go to the midway and the game, you walk up to the game, we're going to play the game. There's an array of 100 cups in front of you, and you have a box of 100 ping pong balls. The game is simple. Throw your ping pong balls up and get one ball in every cup. Seems simple. Are you ever going to win that game? No, you're not. So Our bulk sample is that box of ping pong balls, and the microchambers are our cups. And when we throw them all up, it's a random distribution going into those microchambers. What you end up with is getting a whole bunch of empties and a whole bunch of ones, and you're going to get a few twos, threes, and fours. That's what you actually get. That feels right. We kind of have a real idea of what that system would look like. And that is the kind of data we generate in digital PCR. So it doesn't matter how we amplified each one of those target molecules. We just need to be able to see if there was target in there, yes or no. It's a binary determination when we're done. This microchamber had nothing. This microchamber had something. We take that nothing and something, and we can assign a distribution of the total bulk sample concentration. It's a little of a mental leap, but that is what people call the Poisson distribution. We know how many are empty and how many are full. And in this situation where we had 100 cups and 100 balls, the idea, and it feels right to say about a third are empty and about a third have one, and the remaining third is the twos, threes, and fours. The Poisson statistics and the Poisson distribution is that measurement of how many twos, threes, and fours you have. So if you can measure something and nothing, you can apply all of the Poisson statistics. So that's what the software actually does, is it generates yeses and nos. So again, if we're talking about efficiency, it doesn't matter how fast or slow we get to the end. We just need to get to the end. And that is when the system actually collects data, it collects a pre-read at 0 and an endpoint read. That's all the data it collects. It just does it in bulk. And it's the crunching of those positives and negatives that actually will be able allow you to generate a concentration number. So it's using the same nuts and bolts, the same chemistry type to generate a different kind of data. That's a 30-second. What is digital PCR? Sorry, did I answer your question? Yeah. Okay.
Anyone else? Okay, awesome. Let's use it one more time.
Let's go eat.
Yeah, it's just like Susan said, now's the time we eat. A quick message from Lenny, who actually want to give a big shout out to Lenny Bain, a really helpful Thermo Fisher contact. He's the actual producer of this event. This is a Lenny Bain production here. He wanted me to remind you all that he will send out links to these presentations in the next couple days or so. But in the meantime, there's Qdoba waiting for you in 1360, which is just turn around the corner to the right. Help yourself. And if you have any questions about Thermo Fisher things, you know who to talk to, or BioGarage things, come find us. Thanks for coming out.